happy that we are back here. So I welcome you all back for another few hours of entertainment, discussion, and exciting knowledge and information about what's going on out there in the real world. So we will have first Rene Spijker, who also has an interesting background because he's been working in academia, has published lots and lots of work, including with some of the people who I see here on my screen, live at the party, for example, one of them. But you have a foot in both academia and industry, right? And so that seems to be a common thing that I observed today. So Paul, who might have left by now, I think he had to leave, is one of them. So I would say, without further ado, I hand over to Rene, who will be talking about systematic reviews and related problems.
Okay, so let me see. Yep. So let's go to all the way to, so you've seen my presentation.
That was a bit fast. Could you do the one again?
You see my presentation now? So this is it. So what am I going to talk about? I'm going to talk about systematic reviews, and I'm going to talk about systematic reviews during the pandemic. My name is Rene Spijker, and I do have some disclaimers, I'm calling them these days, so I do work in Amsterdam as an information specialist within a medical library where I support systematic reviews. That's about half my time. The other half of my time, I'm back into the city where I'm also living in. That's Utrecht, that's in the center of the Netherlands, where I'm both a senior scientist and a staff member for Cochrane Netherlands. Besides that, I'm a member of the Cochrane Information Specialist Executive and the Cochrane Council. And what Cochrane is, I'll be explaining in a minute. I'm a member of a couple of methods groups or methodology groups, which is the Information Retrieval Methods Group, Screening and Diagnostic Test, Prognosis, Rapid Reviews. Also on board as a member of the International Collaboration for the Automation of Systematic Reviews, which we'll talk about later on as well. I have been part of the CLAY eHealth, which is one of the information retrieval competitions here in Europe on the technology assisted review labs in 2017, 18 and 19, together with Evangelos Kanoulas and Life, who I thought I noticed sitting here in this meeting as well. I do some work for the BMJ Evidence-Based Medicine and for FBRES, which is a European Union funded project. I'm a co-lead for the efficiency in systematic reviews. These are all disclaimers, so whenever you see some of my work passing, it's because I'm biased. So what is a systematic review? And it's a very good question because a systematic review is a means to an end. And the means is that you gather information in such a way that you try to minimize bias in all your steps as much as you can. And to do so, we have a very systematic and explicit methodology matured over the years within Cochrane. And I'm mentioning Cochrane here because Cochrane, which is a medical, which works within the medical domain, is one of the first organizations that started to make systematic reviews be accepted more broadly. And what you initially do is that you gather evidence from studies that are being published or unpublished studies. You gather those, you select the studies that really do matter to your initial question, and you have a very explicit question. It needs to be very focused, the question. After you've selected those studies, you do extract data from the studies, and you need to extract that data to do one, use it to determine the validity, which is we call risk of bias assessment, to see how well the methodology was and whether or not you could draw the conclusions that you did based on the methodology that you use and how well you followed those methodologies. You need the data that is being extracted to reuse it. And that's exactly what we do in a systematic review. We reuse information from primary studies to collect all of them. And to make that more informative to you, so on the left-hand side, we have the Cochrane logo. And the Cochrane logo is what we call a meta-analysis. And you can do a meta-analysis if you just have two studies. And the two studies, you just pick them and you do a meta-analysis and you get what we call an averaged effect. And that's really nice, but it doesn't tell you whether or not that's a true reflection of the true average effect size. So we needed something around the meta-analysis that could give us some validity of that we were capturing the true effect size. So what am I talking about? So if you look at this, this is what we call a forest plot. This is for every single study. We've looked at in this case, the odds ratio, but it can also be an effect size. And for every study, we plot that against an X and a Y axis. You see here one study, you see a difference in confidence intervals. Quite often this has to do with the number of included patients. And what you can do in the end is to plot everything and then do a meta-analysis across all of these studies. And the more studies that you have captured that have been done, the more meaningful this meta-analysis is. So if I would have just picked two studies, because I had them in my drawer, I would have done a meta-analysis, it wouldn't be as meaningful as if when I've done a thorough job of locating all of the evidence that is out there. And this represents an effect size. And why was this so important? Well, up until this moment that people did this meta-analysis, there were a lot of children who were born prematurely and who were dying because they didn't get any treatment. And there were a lot of studies already out there for many, many years that looked at corticosteroids and the usefulness of that for early born infants. And they couldn't really see an effect size. And it was only when people started to look at all of the evidence, so this is what's been done here, that we saw that there was an effect size, It was just not a very large effect size. But given the number of children that are being born prematurely, then even such a small effect size is a lot if you start treating people, pregnant women with corticosteroids for the development of their premature child. So, and this was such a, let's say, breakthrough that people all of a sudden saw, well, okay, hang on a minute, we shouldn't always focus on just looking at a single study, but we should collect all of the studies together and look at the results across all of these studies to have a proper understanding of the true effect size, which is very meaningful for practice, that people started to see the value for that for everyday decision making. Well, this coincided with something which we call evidence-based medicine. Evidence-based medicine doesn't mean that the evidence is leading, it's always telling you what to do, but the evidence is there to inform you about the decisions that you have to make whenever you do clinical work. But these days it's much broader because it's even used to determine policy making. So what treatments do we do, but also what are we going to fund for research and what are we not going to fund for research. So systematic reviews are being used more and more even outside of the medical domain these days, so in legal, environment. We see a lot in the global issues at the moment. We see even in computer science, we see systematic reviews popping up. So it's broader than that. So if we now look at a whole body of literature, and I was just looking for the literature, but randomly I would miss certain studies. If I'm missing studies randomly because I cannot capture everything, then that to the end result wouldn't make that much of a difference. It will make a difference in the broadness of the confidence interval, but for the overall effect size, it wouldn't make that much of a difference. However, if due to my searching or the selection of the studies that I'm including, I would make a systematically error. So for instance, in this case, I'm only looking at, let's say, high impact journals. And I'm not looking at the rest of the body of literature. And only the high impact journals would give me such a result as I'm seeing here. Then the average effect size is all of a sudden shifted, so it's biased. So in this case, for the systematically missing of literature is a real issue. So this is why we always try to capture as many studies as we can. And this is, we just had it in the beginning, where most of the emphasis these days when searching is on precision, our emphasis is not as much on precision, but it's more on recall. And to go back to one of the, I think it was one of the NIST, so the track trials, which was called Total Recall. This is exactly what we need. So we try to identify every single study that's out there. We do appreciate that it's hardly possible to find every single study that's out there, because that's a very difficult thing to claim in the end, but you at least try to find the majority of the trials. So before we had the pandemic, there was already this conception of that the publication landscape was shifting. And back in 2010, already 11 years ago, Hilde Bastian did an investigation, and she saw that if she looked at PubMed, which is one of our major databases, she saw that about every single day, 75 clinical trials were being published. And if she looked back 6 years later, that already increased to about 100 clinical trials every single day. So that was already a huge infodemic, as we would call it. There were also multiple systematic reviews coming out, and not just for interventions, but there were also systematic reviews on prognosis, diagnostic test accuracy, qualitative ones, evidence maps. The new areas for systematic reviews were being used, so not just in treating patients, but also in policy, the research agenda, research waste. So this meant that we were dealing with a huge increase in the amount of data that people were publishing, and a huge amount of data that we needed to shift through. And because it's a highly manual process, and we come to the process later on, the average time it takes to do a systematic review varies from 6 months to 3 years. And in the old days, 6 months to a year was something that for some issues was acceptable, but sometimes we had policymakers coming to us and they wanted to have an answer within a couple of months. So that highly manual process, which is very much aimed on reducing bias, it becomes unsustainable. And this was even before the pandemic. Yeah. So this led in 2015, during a Cochrane colloquium, that a group of people came together. Paul Glaziou and Per Van Wyk were the ones who initiated this, where they stipulated, well, if this is going to be the trend, and we think we're in an exponential phase, so it will just increase multiple times over, then we need to start preparing ourselves for this. And this is the Vienna statement, says that the systematic review involves multiple tasks, and each of them has their different issues. So all of these issues need to be addressed. Automation may assist with all of these tasks, or some of these tasks, and we need to identify which of these can be assisted with automation. The process for each task can and should be continuously improved to be more efficient and more accurate. So we shouldn't stop once we've improved something and then leave at it, but keep on going. Automation can and should facilitate the production of systematic reviews that adhere to high standards for the reporting, conduct, and updating of rigorous reviews. And this is very important. So automation can never lead to a lower quality of the systematic review, because people will never trust the systematic review anymore. Development should also provide for flexibility in combining and using components. And what we meant with that at that time, so if one group, let's say in Seattle, is developing something, another group in Canberra in Australia is developing a tool, and they both are being used within the total process of a systematic review, they need to be able to talk to each other, because if I'm doing one task with tool number one, and I need the outcome of tool number one to do the task that is being assisted by tool number two, it shouldn't interfere too much with the workflow. So different groups with different expertise are working on different parts of the problem. So this is why we meant that we need to gather more people than just people from the clinical domain. Well, we need computer experts, we need methodological experts, we need topic experts. All of them need to come together to get this started. Every automation technique should be shared, preferably by making the code and the evaluation data and corpora available for free. So that means open science. And all automation techniques and tools should be evaluated using recommended and replicable methods. And the evaluation is coming in here is to know how reliable such a tool is in performing its task. So what is the systematic review? Well, a systematic review, because it's so systematically, it has very discrete steps. So we can draw these, and this was from a publication by Guy Zapnut in a systematic review 2014, where all of these tasks were being split up between the preparation, so the preparatory work, the retrieval phase, synthesis phase, and the write-up phase. So what is in the preparation phase? Well, that is, you need to formulate your review question. Sounds very simple, but quite often this is a very difficult task to get to the real core of the question that you really need and want to answer to inform a decision. You need to identify whether or not people have already done a systematic review for that question, because if people have already done it, then doing another one is a bit of a waste of the research. Next, we're going to write a protocol. The protocol is there for a reason. It's because everything that we do subsequently cannot be changed. Because if I'm going to look at the evidence and then change my methodology accordingly, I'm going to introduce bias, because all of a sudden I'm aware of what there is and I might steer myself towards a certain answer. So that's why I'm doing that explicitly upfront, so in a protocol. After I've done that, I'm going to devise a search strategy. And a search strategy is both where I'm going to search, so what databases, where I'm going to get my records from, and within a database, the search query, as most of you know, how I'm going to search. Then I'm going to perform that search, quite often in multiple databases, because, for instance, PubMed, which is our core database within the medical field, only holds about 10% of all the information that's out there in the biomedical field. So 5,000 journals are being indexed in PubMed, while we are guessing at the moment it's about 50,000 biomedical journals in total in the world. Because there's overlap between several databases, we need to deduplicate. Sounds easy, but data between databases is not always similar. So even though it's the same publication, the metadata might be different. So the way the journal has been written out, the abbreviation of the journal, maybe the authors were just turned around, first name first, last name second, and then in the other database, the other way around. So deduplication is something that needs to be addressed. After that, we do manually screening of the search results. So first we do, because we have a problem within the biomedical domain, and that is we do not always have access to all the full text, we first screen on what we call the most obvious metadata, which is title and abstract. And the abstract, in this case, is an abstract written by the authors of the full text paper. So we screen on that and then determine whether or not it's potentially relevant for our ultimate question. Of those potentially, so that screening is being done in duplicate, blinded, so that where we reach intersubjectivity, something which we call, so that two people have an opinion about, and when two people have the same opinion about include or exclude, it's going through to full text. Discrepancies are being resolved by discussion or by a third reviewer. Next, we need the full text of a paper. And quite often you do not get all the full text because you don't have a subscription to a journal. So sometimes you can ask other people, but sometimes it's very difficult to obtain a full text copy. Then we're going to screen the full text, also manually, also in duplicate, blinded again, to put them against our inclusion and exclusion criteria. And those reflect whether or not they are capable of answering our initial question. After that, we want to make sure that our initial search really captured all of the potential results that we thought we needed. And so we do a check. And that check quite often is snowballing, is forward and backward citation tracking, to see whether or not there's citations within the final included studies that were relevant as well, but were missed with our study, with our initial search. Once we check that and we're finished with that, we start to extract data, and we extract data from the trial that was conducted. That is something that we need to synthesize our data. So that's, for instance, to inform the meta-analysis, but also the extracted data is there to assess the validity of the data, so to assess the risk of bias within the study. And this is then in the end all summed up with a meta-analysis, if possible, because sometimes trials are just not comparable to each other. Then we just synthesize in a narrative way. We tell something about the validity, and then we conclude whether or not what the end result is on the hypothesis that we proclaimed in our initial question. So these are all the steps that we do in a systematic way for a systematic review. So are there tools for these different steps? Yes, there are. This is wrong. So here are some of the available tools. Those are not the only ones. There's a very nice web resource, which is called the Systematic Reviews Toolbox. And there's already a couple of 100 tools for the different areas within a systematic review. So some of them you can see here. So there's some for searching, some for snowballing. There's some for extracting data. There's some for helping you to write up the end, to do the meta-analysis. There's also tools for risk of bias assessment. So, however, what we do at the moment is quite often that a systematic review is just a snapshot in time for what we know up until that moment in time. Most systematic reviews published today finish there. So they just give the snapshot of the current knowledge that is out there. Cochrane reviews are a little bit different in that way, because we do accept that topics do change and there's a dynamic view in there, that new evidence is gained periodically. So Cochrane reviews are reviewed every 3 to 5 years, whether or not they're still up to date, and if not, they're being updated. So new studies are being added, the analyses are being redone, and then maybe the conclusions are being adjusted if necessary. Another newer form of that came out of that, although Cochrane reviews can be seen as some sort of a living review, but with 3 to 5 years between them, is a living review. And a living review is something where there's more of an active surveillance of the literature going on, smaller increments, where people are monitoring whether or not new literature has been published, then capture those relevant literature, look at that, and then have moments in time where they decide, well, looking at the relevant literature that we found now, we think an evidence update is warranted. And to get some guidance on that, you can go to there's Living Evidence in Australia, or there's a web page from Cochrane about living systematic reviews, where you can find more information about that. So this is the corona pandemic, which came upon us somewhere, well, for most of us, somewhere in February, March 2020. For some countries, especially China, it came a little bit earlier. And we saw a huge increase in publication. So I just did an update and looked at it from one of our trusted resources, and we had about 200+ references, so studies being published since March 2020. So 200,000. It's a very dynamic field. And with the introduction of what we call preprint servers, so in the old days, whenever you were submitting a paper, you were submitting that paper to a journal. The journal would send it out to peer reviewers. They came back with comments. You addressed those comments, and then after 1 or 2 of those rounds, a final publication came about, and that took a couple of months. what people have done now within the COVID pandemic is that, well, that's taken too long. So we are already going to publish our pre-final edition on the web. The arXiv repositories are very famous for that. So what you need to consider is that there's no pre-review has been done on those publications. So other people have not looked at it. So there's a bit of an issue there. And that's something that we saw a lot during the pandemic, because as soon as a preprint comes out, and quite a few of those investigators also found it very necessary to put out press releases pinpointing towards the preprint server for that just newly released publication. And it became already within the broader domain, whilst people from within their group, from within their peers, have not had a look at that paper and judged it for its quality, and whether or not more data was needed to have a substantial paper. So that gave us a bit of an issue. It also gave us an opportunity, because of these preprint servers, good papers were very early on already known to the people who needed to make the decisions on how to treat patients. So there's some good and some bad going on there. And we need to settle and look back in a couple of months' time or in a year's time on what where the barriers and where the facilitators were for these preprint servers. However, we also didn't have months or years to inform policy, which is something that from a systematic review point of view, we were used to. Now we need it within days or weeks. And especially also with the rapid spread of misinformation, a huge emphasis came on the living systematic reviews, but also on very, very rapid. So a couple of organizations picked it up, and what we have is, and this is just a couple because there's many more. So we had from the Bern University, we have COAP, and what they did is very easy. They just assembled, like WHO did and what the CDC did, to make a repository with a lot of studies in there, so you could use that to inform you. So you didn't need to go to all the various libraries, so databases, to search those databases. They already did that. They already deduplicated, so you could use one single source to inform your evidence synthesis. The other one is something which is led by Epidemiological in Chile. They were already working on this for many, many years, but with the COVID pandemic, they stepped it up, which is the LOVE platform, where they have a platform where evidence comes in and is being labeled. Another initiative, which was COVID NMA, which was first led by Cochrane France, but in the meantime has a huge group of people working together where they do a network meta-analysis of every drug and non-drug intervention of COVID comes around. All of these are still very much dependent on people. Same as COVID EMD, which was kickstarted by McMaster University. And some of them have already started to introduce some sort of automation. And I think out of these, the people from Epidemiological have been the furthest into introducing that. So we're going to talk a little bit more about some of those. So Epidemiological still uses quite a lot of manual work to identify studies, but then do deduplication and some of the pre-processing work using classifiers and using a word-to-vec kind of environment to be able to add metadata to the studies that they find. So they'll be able to pinpoint, well, if you're looking for anything that has to do with, let's say, cetirizine, and it's within, let's say, older adults within COVID-19, then we can already limit the number of results that you need to look for. So this is already a combination of manual and let's just call it for the sake of it, artificial intelligence coming together. So that's machine learning, NLP, and knowledge graphs, but in a very rudimentary form. Epistemological wasn't the only one, because in the beginning, also the Allen Institute also already started gathering a huge amount of information. They got some of the publishers on board to be able to already give them the full publications. And what the Allen Institute did is they attached a lot of metadata, both from the publisher that was already there, so titles, abstracts, journals, whatsoever, but they also did on the backbone, and I think it was BioBird, but it could also be a Skybird implementation to attach more metadata that you could use for your searching. There was still a lot of noise in the base Cord 19 set, which was composed of information from publishers, WHO, CDC, and a lot of that. So this prompted investigators at Elsevier to investigate whether or not they could, let's say, liberate the real scientific studies from the non-scientific studies. And what they used for that was a BioBird-based, so not a bag of words, but BioBird-based, and then used something from XGBoost in an active learning environment where they did multiple rounds where annotation was done, so classification was done by manual experts, and based on the feedback from those manual experts, they trained the classifier in a couple of rounds until after a couple of rounds it was so good that it was very well on its own able to classify documents coming out of Cord 19 into whether it was scientific or whether it was non-scientific papers. Another method of doing something like that was initiated by Cochrane. Cochrane wanted to limit a register only for real clinical studies. So they were only interested in something like randomized controlled trials, cohorts, and observational trial studies. So what they used is crowdsourcing, where they got a lot of people together annotating studies that came in from various resources. So they did a search across multiple databases and resources, and then the crowd annotated it for it being either a clinical study or a non-clinical study. Non-clinical studies were not taken into the study register, and the other ones were then annotated further with what kind of study it was. Was it an RCT? Was it a cohort or another? Was it a cross-sectional study? And was annotated for what domain it was. Was it an intervention? Was it a diagnosis or a prognosis? So this is a registry that you can use and where you can limit some of your results already on very specific, it's an RCT within the domain of intervention. So what can you do? Well, I've been part of a suite of what we call living systematic reviews on diagnostic test accuracy. And what we used is we used both the Cochrane study register and we used the live feed from COAP. The first time, we already started somewhere in April, June of 2020. So the number of results was still quite manageable. So we first screened manually, and learning from that, then we started to build classifiers based on the information that we got from the first reviews, and then tested it out on a subsequent round of manual screening to see how well our classifiers were, so that in the end we could use our specific classifiers per review to update our reviews every single time. And we've updated some of our reviews about 6 or 7 times now with our searches, and we've been publishing 3 or 4 iterations of our systematic reviews doing that. Still a lot of manual work, even with the classifier, because we're so sensitive, because we do not want to miss any studies. The precision is still very low. Even though the recall is very high, the precision is very low, so we need to manually screen still a lot of noise. Let's go to the next slide. Yeah. So then there's also this, can we use in the future, can we use one database instead of all these multiple databases to perform such a feed? And then at the epicenter, so under the guidance of Ian Schemold and James Thomas, Epicenter started to look into Microsoft Academic Graph. They already started doing that before the COVID-19, because already in Chile in 2019, they were already presenting this idea, but they've updated it, and they're using the Microsoft Academic Graph first to do a pre-selection. They're using, and they only looked at this from the viewpoint of that you've already done one iteration of a systematic review and you want to update this. Yeah, so you already have information about relevant documents. So and those relevant documents are then lined against a new iteration of Microsoft Academic Graph. First screening is done on a cosine similarity across the whole graph. And then the end results from that, so that's screening against all 200 million of Microsoft Academic Graph items. And the subset that comes out after the cosine similarity, they can run classifiers that they've built based on this specific subset, so that you have a much limited set of data that you need to screen. And they did a major evaluations on that against a manual selection process from multiple databases. And what they saw is that all the relevant studies were indeed captured with Microsoft Academic Graph. And they even identified studies that were not previously identified through the manual process. So it showed that Microsoft Academic Graph could indeed be a very useful tool in the future. Disclaimer here, Microsoft found its product so successful with all the work that a lot of these review groups have put in. It's not just this one, but Lens.org has done a lot of work using Microsoft Academic Graph, that Microsoft Academic will be closed by the end of this year. They're going to now commercialize this product for other domains with all that they've learned from the Microsoft academic world. It's a bit of a shame, because we were starting to develop our tools based on this. So at the moment, people around the world are now looking into what other resources might be able to replace something like Microsoft Academic. So what we've now focused on was at the retrieval side of the studies, but is that indeed one of the resources, the resource used within a systematic review of all those tasks that we saw, that's the most relevant for us to search for automation? So this is a review that we produced. It's one of a suite of reviews that we do for the FBRES project. And when we did an investigation there, both through a survey and through a literature search, is that we discovered that administration is the highest burden in research use during a systematic review process. Next comes data extraction, so extracting the data from relevant papers, critical appraisal, study selection, and in the end, searching. So searching and study selection are resource intensive, but not as resource intensive as the other ones. Whilst most of the tools at the moment that people are investigating are aimed at both searching and study selection. It's probably because it's the easiest one to address. It's not the one that's going to have the major impact on the whole process of a systematic review that we would want to. So what have people have done already up to this point? Well, as part of the CLAY initiative, where I was part of, for instance, we did a core data set against all different kind of methodologies of doing a screening job, so title and abstract screening, and the other one that we did was not giving a search selection, but just telling people this is the whole of PubMed, this is your question, and in how many iterations can you now find all the relevant documents for this question, and that's what you're seeing in this one, in this graph. So, and what people have used in this one is they used either a support vector machine active learning. Other people use logistic regression active learning. Both of these most of the time were on back of words, but some also used some form of BERT or topic modeling as an input. Other people use learning to rank mechanisms, BMI, 25, for instance. So there were a couple of these, but the one that outperformed most of the times was one developed by the team of Waterloo, which came from the, I think, first challenge was within the legal domain, which was a continuous active learning machine on I think it was a support vector machine classifier. And that did it very well. But this is all retrospective analysis, and all of the things that people have looked at the moment are retrospective and not in a prospective manner. The other thing that people have developed at the moment is something called Robot Reviewer. This is where we do the risk of bias assessment based on a PDF of a paper, which is pinpoints towards which domains are relevant for judging a randomized controlled trial for its bias. And it also does neatly, I didn't put a picture in here as well, but what they also do, they annotate within the PDF, so they highlight the areas where the information can be found on some of the domains where you can find information around bias. Another tool is Dexter, and Dexter is capable of extracting relevant data from clinical studies for further processing. And some evaluations are ongoing on that machine as well. So why haven't we already switched to some of these tools for our systematic reviews? Well, there's some barriers for the implementation. One of the barriers is for the screening process is because we're now not presented with a Boolean search results, which I'll come back in the next, is because we're presented with a gradient, it's very difficult to judge when we can stop the retrieval process. So, and this is subject to some research at the moment. So this is one from Max Callahan, Evangelos Kanoulas at Amsterdam University has had a look at that as well. So can we introduce a stopping criteria? Can we predict when we, for instance, have captured 95% of all potentially relevant documents or not? But at the moment, we do not have something like that really working. Yeah, there's one tool, I think it's the tool from Swift Active Screener, who uses a predictor where it's continuously updated during your screening process until it predicts that you reached approximately 95% of all relevant documents within your search results. But this is still a debate where people are saying, well, as long as we haven't fixed that, it's very difficult to switch over. And this comes to why people are still very much in love with Boolean searches within the domain of systematic reviews. Because Boolean searches, one, they still give a good result, and they work very well across the multiple databases that we use at the moment. But also, Boolean searches are quite transparent in the fact that they at least show you on what kind of decisions an article is being included or excluded from your search results. It either, and I should have spelled Boolean right here, sorry for that. So as all terms in your Boolean search are visible, and we always present our Boolean search query in our papers, people can judge whether or not the search was conducted in a correct way and whether or not it's indeed capable of retrieving all relevant documents. So this is something that we really need to keep in the back of our minds when we switch over to machine learning, let's say AI kind of methods, is that we also need to bake into that system that transparency on the micro decisions of the machine need to be explainable as well, so that we can explain why a certain paper is included in the final result and why it's not. So is there some good news? Yes, there is some good news. The people in Australia, led by Paul Glaziou, they're doing now systematic reviews in 2 weeks' time. They're very, very condensed systematic reviews on a very condensed topic, but still they've been able to do so. And why is that? Well, they've chosen to have a very highly experienced team to use. They use something like a Scrum kind of methodology, so daily brief meetings, use some text mining tools to inform or build the search query, translate their search queries from database to database in a semi-automated fashion, use deduplication tools to deduplicate the results from the various databases, use robot search to select randomized controlled trials out of their system. So this is a classifier trained on identifying randomized controlled trials. And they use robot reviewer to help them assess the risk of bias assessment. So it can be done under specific circumstances, and we can learn from these process innovations. So what do we need in the future? And I think I'm almost there, yeah. So what do we need? Well, for rapid decisions, it's essential to have the most current representation of the scientific evidence. This is what we really learned during the COVID epidemic. Well, pandemic still, although I would call it endemic. Current systems are too slow to meet these challenges. We have a problem because most of us still do not have global access to all full text papers due to paywalls. Most of the journals have still a lot of their scientific articles behind a paywall. Although open access is on the rise, it still not captures all the relevant clinical trials that we need. Most scientific articles are available in PDF and are aimed at being read by a human. So first we need to re-engineer again to liberate the information that's contained within the PDF, and we need to understand the information. So it's still very unstructured, although scientists still think that they are reporting in a very structured manner. For the machine, it's very unstructured. So we still need to make a translation from the unstructured data to the structured data. So what we need is to have a system in the future where the metadata is much more available for one, more specific retrieval. So keeping the very high recall, but increasing our precision. We need to have the metadata available that's needed for the validity of the study. So the risk of bias items being extracted and highlighted. We already need the important data from the article available as metadata to interpret it. So this needs a much better combination of machine learning, NLP, and knowledge graphs than we have at the moment. And this is, I think, where I'm going to leave you. Yep, this is where I'm going to leave you. So I'm hoping that we're open for questions.
Thank you very much. So initially you said, oh, I just have a few slides here, and now used exactly the right amount of time. So I'm sure, so thank you very much. I'm sure there will be plenty of questions. Let me perhaps start with one, and that is, you talked about people now publishing stuff on archive, etc. So essentially to get it out quickly, which of course causes plenty of problems for you. Now, I recently came across a study, actually last week, where they found that it's actually worse than that. So there's this journal called Naunin Schmiedeberg's Archive of Pharmacology, which goes back to 1800 something. And they found that a lot of the studies that they that passed the peer review process were fake studies, mostly from China, because of the pressure for people to publish stuff. And so it seems like some of these were retracted. Most of those were retracted, but this is perhaps just the tip of the iceberg. So do you also see problems like that affecting the work of a person doing systematic review?
So yeah, this is it's a double-edged sword, because whenever we do a systematic review, we are going over the evidence again. So even after the peer reviewers have done so, we discover quite a lot. I mean, this is from personal experience. When you start to do a data extraction, you get to know the papers very well. And so all of a sudden you come across a paper and you start extracting data and you think, hey, hang on a minute, I've seen this data before. And we've already identified quite a few of those studies that I'm not going to name names from countries or whatever, because it happens everywhere in the world. We all have our own pressures, but that all of a sudden that you see a paper using data that was already published by someone else. So we do see that. And sometimes what you also see is that you come across data, and then if you start to look at it, you say, well, this can't be right. So what they are saying here can't be based on the numbers that we're seeing here. Quite often this comes through when we're trying to do the meta-analysis. And because we're comparing different studies, so all of a sudden you see a huge outlier on something. So systematic reviews can help in that process. We are hoping we do not have to do that. So we hope that the peer review process would be a filter for that. But the peer review process is on an enormous stress at the moment. I mean, if I look in my email box, how many invites I get and how much time I do have, I mean, it's almost impossible. So hopefully there will be systems in place that could help on that end as well in the future.
Yeah. Okay, thanks. Yeah, no, that's very interesting. Because essentially what they did, so what they wrote about in this report was a similar thing. So the publisher was eyeballing some of these papers and realized some of the tables looked the same, and then they started to drill down and found that a lot of this was actually dodgy. Yeah. Okay, thanks. So Tony, I knew Tony would have a question. So over to you.
Yeah, hi. Yeah, it's a great talk, Rene. Really enjoyed it as always. I've got a question. It's a sort of, I'm not sure whether it's a philosophical one or not, but you know, you were talking about the publishing industry, and I know there's a lot of need for change around the business model and so on. But I'm thinking more, sort of following on a little bit from your last point, because a lot of the inefficiencies that we see both in publishing and the subsequent systematic review is the fact that we publish essentially to impress our peers who are human beings. And so part of that is, you know, writing it up as with a narrative in a PDF, and part of it is just representing it in a form that articulates the contribution rather than results and so on. And I'm just wondering whether, in your view, there's a kind of vision for how the publishing industry might change to be, I don't know, I'm struggling for the right metaphor, less of a push model and more of a pull model, where perhaps part of that acceptance process is not just publishing the manuscript, but populating the relevant database with the relevant results. And obviously I'm waving my arms here because I don't know what the relevant database would be or the relevant results would be. But you see where I'm going with this thought, where we just cut out all that fuzziness in the middle, where we've got to take stuff that was originally structured and became unstructured, now put the structure back in it. And it just seems incredibly inefficient.
Yeah, so I do not have the solution as of yet, but I do know there's a couple of initiatives that do address this. I mean, there's the new European initiative that's going on clinical studies. So this is within the clinical domain at the moment, but whatever trickles down to other domains, I'm sure, is that first you need to register your trial before you even conduct your trial. So we call this clinical trial registries. But those clinical trial registries now also have to attach some of the core data that came out of such a trial. So this is the raw data. So we are moving more and more on liberating the raw data that's underlying the publication as well. And what people are now doing, and I know, for instance, Elsevier is already doing that as well within one of their projects, is to link the raw data that they can find within certain university repositories or different repositories such as Zenodo, to link those to the publications so that those two are linked. Because one of the problems for a lot of the data sets that people are publishing is that they lack a lot of the metadata that we need to interpret the data, about the validity, so the methodology and all that. So we need to have that link with a publication where people are telling what it is. So there's already initiatives ongoing where people are trying to link that, but it's also something something just occurred to me last week, so it's very fresh, but the publication in itself, we will not be able to change that much because the necessity is still there for a publication to inform human readers about something. So what we need to figure out is how to translate that human information towards structured data, or where structured data cannot be compiled from the publication, how we can add the structured data that the scientists do have within their own computer, and how we can tap into that. So and that's more and more happening. So people are still more and more making their data available, but sometimes it's quite difficult due to legal rights as well to make all data available because of informed consent and all that. So we need to find a form in which you can do that and which you can't. But probably there's some people within the audience who have other ideas about that as well. But this is my understanding. And at least the publishers, well, the one I've been talking to, which is Elsevier, is already aware of this as well. So they are also thinking more into knowledge brokerage than just a standard scientific publication.
Yeah. It's a slow process, though, isn't it? I mean, you rightly point out a few of those initiatives, but I think, is it fair to say they're almost exclusively all in healthcare information retrieval? I don't think any such thing exists in all the other systematic, literature review domains. So,
no, I can. It's always difficult because in some of the legal domain, the availability of full text documents is much easier than in some of the medical domains. And sometimes within the legal domain, you can also build a system which goes into a company and shifts through all its legal documents, and then comes out and says, well, we found this, and then it's done because you only need to look at the documents within one company. And so there's different challenges there. But yeah, so we do have a bit of an issue within the healthcare industry.
Yeah. All right, thanks.
Thank you. We have we have 3 more minutes, so I think we can do 2 questions. So Andy posted something, but let me just ask what Martin sent to me directly, because he's just replacing his central heating. So I think he's not close to mic. So he said that he has seen comments that there's a significant increase in national epidemiological studies published in local languages. And he wonders whether that adds some layer of complexity, which nicely aligns with what he was talking about earlier, but you didn't have a chance to be there.
Yes and no. Depends in, so if the underlying data is all in the local language, then we do have a bit of an issue, although we are getting better and better into aligning that. For instance, within the medical domain, if we matched things to an ontology like SNOMED or MESH, there are translations of those data of those ontologies as well to various languages, so we can make use of that. So we can make use as soon as we've translated it from unstructured to structured data, we can do the translation between languages as well. But for those epidemiological surveys, it's also very relevant that we do know the methodology on how they were conducted, so that we need all that metadata. And at the moment, we haven't established exactly what metadata is needed on raw data to be able to use that. So that's one thing that we still need to figure out as methodologists to compile.
Okay.
Yeah.
Thanks, Andy. My suggestion would be the following: if we just ask Rene to answer your question on the chat window, then we could move on, because we are right on time, and we have another exciting session. So first of all, Rene, thanks again. Excellent.
Thank you. Hopefully next time in London again. I mean, I did enjoy my one time that I was there about a couple of years ago, but
yeah, so let's all hope we will be back in London next year. So, but yeah, I'm sure we will. Famous last words.